I would like to bring up James Santangelo, who is a graduate student in evolutionary biology at the University of Toronto. And James is going to tell us about rapid evolution in urban settings using the white clover as a model.
Awesome. So thanks for having me here to talk to you guys today. So I'm going to tell you a little bit about some of the work I've been doing trying to understand how species evolve in response to urbanization as part of my PhD. So as I'm sure many are familiar, we're part of this new epoch now that geologists refer to as the Anthropocene, in which human beings are having a decisive impact on the state, dynamic, and future of planet Earth. Now, much of these changes are being driven by urbanization, in which huge swaths of Earth's natural landscapes are being converted into human settlements like towns, villages, and cities. And prior to 1990, the traditional view of cities was that they were very much anti-life. Looking at a dense, smoggy metropolis like this, how could it support levels of biodiversity that would make it interesting or worthy of study from a biologist's standpoint? And this is a serious problem because, of course, urban environments are distributed across the world. As of 2008, the majority of the human population lives in cities, and urban environments continue to expand across the world. And so there's a pressing need to understand how species are evolving or adapting in response to these changes, and whether we're going to be able to recover some of the species that are most at risk. And so an emerging view is that cities are living laboratories of biological investigation. And I think this view is emerging for three reasons. The first is because of the scale of urban environments that allows us to use independent cities peppered across the world to test interesting ideas in evolutionary biology. The second is because work by urban ecologists since the 1990s have found that rather than decimating biodiversity, cities harbor unique biological communities and altered ecological interactions. And so we know a lot about how cities are influencing the ecology of species, but we still know very little about how they're influencing the evolution of species. And finally, relative to some of the other stuff we've been hearing about today, urbanization is a really recent phenomenon. In the Americas, for example, it dates back but a few hundred years. And so any evolution that we see in cities is really rapid evolution that prior to a few decades ago was thought to be impossible. And so we now recognize that evolution in city could happen in four main ways, which I've broken up here into non-adaptive and adaptive mechanisms. So the first, of course, is mutation, whereby, for example, urban pollution causes individual mutations at the level of individual base substitutions in the genome of organisms that are living in cities. This is the one that we know the least amount about. The next is gene flow, or the exchange of genetic information among populations. As cities become erected and we put barriers in place like roads and buildings, this limits the exchange of genetic information among populations of species that refuse to cross those barriers. And this can have really pronounced effects on the evolution of those species. The next is genetic drift, where you have an ancestrally large population with a ton of genetic variation that then gets fragmented into a small population because of the construction of a city, and that small population now contains only a small subset of that initial variation. And then finally, we have the mechanism that most people are familiar with, natural selection, or the differential reproductive success of individuals leading to the survival of some at the expense of others. Throughout my talk, I'm going to focus on these three mechanisms here. And I'm going to do so using the model system white clover that's been the focus of my PhD. White clover is an excellent model for understanding the impacts of urbanization on evolution because its distribution overlaps largely with that of humans. You'd be hard-pressed to go anywhere in the world where humans are found and not find white clover. And this is because of its history in the agricultural industry of being seeded into agricultural pastures for use as forage and the like. And so it occurs on a global scale. I'm going to focus on a single trait, which is the production of hydrogen cyanide. This is a trait that's primarily thought to be involved in defense against chewing insects. And its populations of white clover are polymorphic for hydrogen cyanide, which is to say that in any one population, some individuals are going to produce it, whereas other populations are not. And this is going to become relevant. And it's worth taking a minute to study the genetics of cyanogenesis. And it's relatively simple from a genetic standpoint relative to some of the other traits we've heard about today. We have two genes. The first gene encodes the production of cyanogenic glucosides. You either have a functional copy or a non-functional copy, and I'm abbreviating this gene as AC. We then have another gene which encodes the production of a hydrolyzing enzyme, linamorase. And again, you could have a functional copy or a non-functional copy. To produce hydrogen cyanide, individuals require a functional copy at both of these genes. If an individual lacks a functional copy at either of these genes, they lack the ability to produce hydrogen cyanide, and they're what we term acyanogenic. Now, this phenotype has a long history of study in evolutionary biology, dating back at least to 1950s, where we found continental scale patterns in the frequency of this trait in Europe, North America, Japan, and New Zealand, such that in the southern warmer regions of the continent, you get really high frequencies of hydrogen cyanide that gradually taper off as you move north across the continent, such that in the north of Sweden, there's very little frequencies. And the change in the frequency of this trait across a spatial gradient is what I'm going to refer to as a cline for the rest of this talk. And this cline is thought to arise because of the cost of producing hydrogen cyanide in really cold environments, where frost causes the cells to burst, resulting in hydrogen cyanide being released in plants that produce it and the inadvertent poisoning of that plant. And so in really cold environments where frosts are prone to breakage, or cells are prone to breakage due to frost, there's active selection against individuals producing hydrogen cyanide, explaining their lower frequencies in the north of Europe. And so what we're interested in understanding is whether these same processes happening at a continental scale could be happening at the more local scale across cities, because of this phenomenon known as the urban heat island effect, where we know that downtown, for example, in Cambridge, where we are now, it tends to be quite a bit warmer than outside of the city in the more rural regions pictured here. And so if the same processes happening at a continental scale are happening on a local scale, then we might expect something like this, where if we plot the frequency of hydrogen cyanide on the y-axis and the distance to the urban center on the x-axis, we'd expect higher frequencies of hydrogen cyanide in the urban core, this would be the equivalent of Southern Europe, and lower frequencies outside the city, the equivalent of Northern Europe. And so a previous master's student in the lab, Ken Thompson, was interested in testing this hypothesis. So he went to 4 large North American cities, Montreal, Toronto, Boston, and New York. In each city, he collected plants from 40 populations along an urban to rural transect. This is the one spanning South Boston here. And in each population, he quantified the proportion of plants that were producing hydrogen cyanide in any one population. And he was interested in understanding how the proportion of plants changes as you go from inside to outside of the city. And what he found was the complete opposite that we predicted, which is often really interesting in science. And so we find that inside the city, you actually have much lower frequencies of hydrogen cyanide than you do outside the city. Remarkably, he found this same pattern in 3 out of the 4 cities that he assayed. Now, he tested a bunch of different mechanisms that could explain this pattern, but ultimately settled on urban to rural snow depth gradients as being the main cause. And the interesting thing here is that in the winter, the urban heat island effect melts all of the snow on top of the plants. And so plants in the city are then exposed to the frigid air temperatures, resulting in selection against plants producing cyanide in the same way that we see in Northern Europe. Incidentally, the one city that didn't show this pattern was Montreal, which gets so much snow that no urban heat island effect can melt all of it, and so there's no pattern in Montreal. Pretty clear cut. There's a problem, though, which is that one, he only had a few cities, and so we don't know how general this pattern is. And the second is that all of those cities get snow. And so we don't have a control case. What's happening in cities that never get snow or never see frost? So I'm going to address the following 3 questions for the remainder of this talk. The first is, how repeatable are these clines across urban and rural transects along a latitudinal gradient in eastern North America? The next is, can we predict the presence or strength of these clines based on regional patterns of snow depth or minimum winter temperature? And finally, are there any other mechanisms that might explain the presence of these clines across cities? So I'll start with the first question. At the start of my degree, what I did was I went to an additional 12 cities in eastern North America, all the way down to Tampa, Florida, in each city following the exact same sampling protocol. And so to show you some of that data here, we have, again, the frequency of hydrogen cyanide on the y-axis and the distance from the urban center. When we look across all 16 cities for which we have data, the pattern is clear. There tends to be, on average, less hydrogen cyanide inside the city than outside the city. But there's variation in this, and this is averaged across 16 cities. It turns out that 8 out of the 16 cities showed this pattern, which is quite remarkable because we never see the pattern in the opposite direction, which is starting to suggest that there might be something going on here. And I'm going to show you some of that variation in a minute when we ask whether or not we could predict the strength or presence of these clines based on regional winter conditions. If we could, it might look a little something like this. Again, same figure that we've been looking at. In northern cities where it tends to get really cold, we would expect really strong clines. But in more southern cities where we don't have that selective agent, namely frost, we wouldn't expect to see it at all. And so we would expect the strength of these clines to decline as we go south towards the equator. If we look at the data, here's a map of winter temperature on the background, all of the cities highlighted here. What we find is that in the north, we indeed do get strong clines. This is the one in Toronto, consistent with the hypothesis that minimum winter temperatures could be important. But of course, we actually see some of our strongest clines in some of the southern cities. And so it's starting to look like something else might be going on here that we can't quite explain yet. And so what's driving these clines? So far the data suggests that frost and snow can't be the only mechanisms because we see strong clines in cities that don't get snow or don't get frost. They could be important in some of the cities, but it's clearly not true in all cases. We know that cities vary in a lot of environmental conditions, and so it's going to be important to try to identify what some of these drivers are. So I'd like to take a minute to think about what other explanations might drive the repeated evolution of the same pattern across space. And I want to take a minute to emphasize the role of neutral evolutionary processes because we've been talking a lot about natural selection today. But it turns out that neutral processes operate quite heavily in cities because of the small population sizes where genes are fluctuating randomly and tend to be lost or fixed in small populations with greater probability. And so remember, the genetics of this trait requires that you have a functional copy at both of these genes. That means if both of these genes are fluctuating randomly, then for this trait to go up in frequency, both of the genes have to go up in frequency in tandem. If either of them goes down in frequency, then the frequency of the phenotype is going to go down just by chance alone because of the fact that you need both copies in a population. And so you could imagine a scenario where you have a rural population at equal frequencies of both of these genes. As it colonizes the urban center, random fluctuations at either of those two genes are going to result in the preferential loss of hydrogen cyanide, potentially explaining why we see less hydrogen cyanide inside the city. So we tested this by developing theoretical simulations of urban evolution, the first of its kind, published in a special feature not that long ago. What we did was we said, what if urban populations tend to be small? And this is consistent with much of the literature because of urban fragmentation breaking up the landscape, whereas rural populations tend to be really large. We allowed the exchange of genetic information between populations, and we were simply interested in how often do we get the same pattern that we saw across space, just by neutral processes without invoking selection. And we see that under really strong gradients in drift, so when there's a really big difference between the size of urban and rural populations, you actually get, on average, quite strong clines in the same direction as the ones that we saw, suggesting that genetic drift on its own could generate the repeated loss of hydrogen cyanide inside the city without ever invoking natural selection. As you decrease the strength of drift, you get virtually no clines, which is to be expected because the populations are the same size across the whole transect. But of course, as you allow more and more genetic information to be exchanged among populations, looking only at the strongest clines, you see a gradual decrease in their strength. And so this alone suggests that the interplay between genetic drift, which is important in small populations like those in cities, and gene flow could structure some of the clines that we're seeing across space. And so to come back to those three questions, the first, how repeatable are these clines? We see them in 8 out of 16 of the cities that we've surveyed. We know that frost could be important in some of these, but probably not the only thing going on because we have really strong clines in some of the southern cities that never see frost. And then genetic drift can cause these clines on the same order as those that we see in the cities. But we have recent evidence that suggests that it doesn't. And I'm not showing that evidence here, but we have evidence from molecular markers, for example, that shows that in the case of clover anyways, there is no increased strength of genetic drift in cities. And so taken together then, all of these things point to natural selection as being the main driver of the urban to rural clines that we see across cities. Frost might be one of the mechanisms, but of course we see this pattern in cities that don't see frost, so we have to start exploring some of the other agents that might be responsible for driving these patterns. And so what we really need is more replication, so more cities, and we need these cities to vary in environmental conditions in ways that might allow us to predict some of these patterns. And so I'm excited to say that we've launched the Global Urban Evolution Project, which you could read more about at this URL. And there's a fantastic group of people that are sort of running the scenes behind the project, ensuring that everything runs smoothly. But I want to highlight the work of 550 or 70 collaborators from 200 cities around the world who are collecting for us 160,000 clover plants from about 7,700 populations. I want to emphasize that a lot of these collaborators are using this as an opportunity for outreach. They're engaging high school students, undergraduate students, which I think highlights the power of urban evolution to engage with local communities and teach people about evolution. We're going to be doing some phenotyping, so the hydrogen cyanide assays I mentioned, but also imaging and remote sensing of all of these populations, in addition to generating genome-wide data to get a sense of how cities are driving evolution across the whole genome, rather than just a small portion relating to the hydrogen cyanide phenotype that I talked about today. The key questions here, one, do species convergently adapt to cities across the world? So do cities cause the repeated evolution of a trait in a single species across the planet? And second, what are the environmental predictors of that convergent evolution on a global scale? And just to give you a sense of what this looks like, every point here represents a city that's agreed to collect clover for us, so we have pretty good global coverage, I would say. People are really excited about this. They're sending us pictures every day on the Twitter. And so it's just really great to see people sending us pictures of them doing field work, and the data sets continue to flow in on a daily basis. So pretty soon we're gonna have an answer to this. And I think projects like this really highlight that cities are living laboratories to study evolutionary change because of their global replication, and especially in cases like white clover, where we have the same species occurring across the planet. And so I would say that not only is urbanization a global phenomenon, but we know now that urban evolution generally is a common phenomenon. And all of the silhouettes here represent clear-cut cases where cities have driven the evolution in one way or another of the species that inhabit them. And the goal is to, as we accumulate our knowledge of how cities are influencing the evolution of life around us, we could start incorporating this knowledge into the design of more sustainable cities and hopefully get a better sense of how this rapid environmental change across the world is driving changes in some of the world's most susceptible species. And with that, I'd like to thank my two labs, the University of Toronto, and of course, all of the organizers for inviting me. I'm gonna flip it back to the picture here because it's more interesting. And yeah, I'd be happy to take questions. Thanks.
James, from the first time I read about your work in the New York Times till today, you did not disappoint. The white clover rules. So there's a question, yes?
Hi, I was wondering if geological strata or building materials are ever a variable in this at all?
Wow, no, not yet. In what sense? Are you thinking that particular strata might?
Well, like how it affects soil composition and then how plants might adapt, not adapt, mutate.
Oh, yeah, that's an excellent question. We have a student in the lab who's going to be approaching that. We don't know much about what's happening to the soil in city relative to outside of the city, but I really can see it as being important, especially because white clover, and I didn't mention this, has a symbiosis with a particular microbe in its roots that fixes nitrogen. And so you could imagine if soil conditions change and it influences the ability of white clover to sort of form a bond with this microbe in the soil, then that could have potential impacts for how it evolves in cities. But we don't have that data yet. But that's a really good question.
Yes.
In your southern cities that show the northern cline pattern, did you correlate that with the past winter, whether they had snow or other bad weather?
Yeah, that's a really good question. So part of the problem is that we're trying to predict these present clines with relatively coarse regional environmental variables. And so we actually need to do quite a bit of a better job, I think, of capturing the difference between cities and outside the city. In that particular case, the data comes from BioClim, which is averaged from 1950 to 2000. And so it sort of incorporates some of those past conditions as a single point variable. But yeah, we do need to do a much better job.
Do you have any idea how fast these things vary? Because if you have like snow in Atlanta last year, will that change the population in the spring?
I think it could. We know now that evolution can happen within a few generations, provided that the strength of selection is strong enough. We don't know whether that's the case now. To answer that, what we really need is temporal sampling. So not only sampling along a spatial gradient, but sampling the same locations at repeated time intervals. And we haven't done that yet.
And yes, related to that, that's a question I had in mind. So the temperatures now are just so extreme in all parts of the countries. You would predict that that's going to have some impact. You just don't know what the impact is, because that's right. So the idea is to baseline these white clovers, so to speak, and then go after them after extreme weather changes, or, I mean, is there a biorepository with all this information? Where do you keep this information?
Where do we keep the data on the white clover specifically? Well, right now it's being held primarily by our collaborators or in our freezer at the University of Toronto.
Okay.
To get a better sense of how quickly some of these populations could respond, it would require going back into the field, collecting more tissue from these populations, and doing that over enough years that we could get a sense of what is the change in the frequency of hydrogen cyanide from one year to the next.
Right, so you would have some data on the impact of moisture and humidity on that phenotype.
Yeah, but really right now only from remote sensing or whatever regional variables that we could get access to. One of the downsides of satellite data, of course, is that it's only part of the picture. It's only environmental data.
Right.
I suspect that some of the things that are driving these patterns are actually species interactions, changes in the amount of insect damage as you move from outside to inside the city, which we know is more important at southern latitudes and could explain some of these stronger clines. But of course, you can't get insect-plant interactions from satellite data.
Right.
And so hopefully, I could convince somebody to pay to send me down there and collect some of that data.
Well, that's great. I'm going to close it to the 5-minute question round, and then we're going to open it up to a full Q&A of 15 to 20 minutes. And I invite all the speakers back up, please. Sharam, Alicia, Katie.